Thank you for the introduction. I will say I'm the lone single-cell DNA sequencing talk in this session, but I do have a little bit of single-cell RNA on the second to last slide, so stick around. So I'm going to be talking about cancer data, and this is the slide that everybody puts up for cancer. Cancer is an evolutionary process, which is defined in many ways by the process somatic mutation. Where a tumor starts with a single cell that's acquired some set of mutations. These mutations range in size over all genomic scales, from single nucleotide variants, changes of one base, all the way up to whole genome duplications. And as cells grow, they'll acquire further mutations, and this results ultimately in a fairly heterogeneous tumor. Single-cell DNA sequencing has been very useful in recent years to help understand the heterogeneity that's present in a tumor and how it relates to tumor evolution, the evolution of resistance, and other useful things to understand. Today I'm going to be talking primarily about a technology called DLP, which is an ultra-low depth single-cell whole genome sequencing data. But essentially, there's nothing in this talk that's specific to this technology or to DNA even. It could potentially be applied to other modalities as well. So with DLP data, we measure hundreds or thousands of cells, but we get very low coverage, so typically less than 0.1x coverage per cell, often much lower. And this data is very well suited for identifying copy number aberrations, so amplifications or deletions of large regions of the genome. But today I'm going to be talking about primarily single nucleotide variants. And the natural question would be, if we have such low depth data where maybe we'll get one read per variant if we're lucky, why would we even look at SMBs in this type of data? And this project really arose out of necessity in many ways. Like going into my postdoc, I knew that I wanted to study, apply population genetics methods to study fitness and selection models in cancer and look at mutational processes and how they shift over time. And ultimately, as it turns out, what we need to do that is a lot of SNVs at fairly low frequencies. And looking at targeted or whole exome data is just not going to be sufficient. And well, we need to do it with fairly high precision, which is where this talk comes in today. Because the challenge is there are really no variant callers designed for low-depth single-cell sequencing data. There are several callers that have been designed for exome or targeted data that really demonstrate that having variant callers that have single-cell specific error models is really beneficial, but nobody's really attempted to do this for very low-depth data, so we typically fall back onto what we've been doing for the past decade or more, and just using standard bulk variant callers. So essentially, we pool all of our cells into a single pseudo bulk BAM, so tossing out all the cell-specific information. We run it through our standard variant calling pipeline, and then afterwards, we have these variant calls where we can start mapping them back to cells or clones. But what we find when we do this is that there's often fairly low agreement between bulk and single cell variant calls. So this is an example of a real tumor that I'll be using throughout this talk, where we have matched bulk and single cell whole genome sequencing data of fairly comparable overall coverage. And we ran both of these modalities through MuTech2, so a bulk variant calling method. And what we find is that there's relatively little agreement between the two, and that there's challenges in both precision and recall. Now, these aren't measuring the same exact cells, so we might expect some amount of real biological divergence, but this is much higher than we typically expect to see from the same tumor. This specifically, the lack of precision here was really overwhelming any real signal in this data at low frequencies when we tried to work with it. So while the ideal approach might be to actually design a full variant caller for this data, that could take a very long time, and we wanted to try something simpler first of just trying to do refinement. So can we take a set of variants that were already called by some variant caller and then filter them down in some way to improve precision? So the standard basic approach to variant calling refinement is just manual inspection. Somebody sits in front of IGV for a long time and picks out artifacts. And as it turns out, people are actually really good at doing this by eye. There are a lot of shared features of artifactual variant calls. They're next to indels, they're near the ends of reads, and so on. But the problem with this, as you might predict, is that it really doesn't scale. When we have whole genome sequencing data, we're talking thousands or tens of thousands mutations, and we can't really have somebody sit and go through all that. But it does motivate the next approach, which is using supervised classification to try to identify artifacts. The last 20 years have shown us that machine learning models are often good at these sorts of problems that people are intuitively good at, but can't really formalize the solution space. And there was a method introduced about 5 years ago for bulk variant call refinement from a group at WashU. And the challenge with any supervised classification is where do the initial training labels come from? And this group put in a whole lot of effort, and it was really a monumental effort in this paper, where they had an estimated 585 hours of manual labeling by trained experts to train this model. And what we found that it works very well for bulk sequencing data, but as we try to apply it to single cell data, as we try to apply it with like slightly different pre-processing pipelines, the performance drops to some extent, and it could potentially be improved by doing more manual labels, but that's not really a scalable approach. So what I'm going to present today is instead an autonomous approach to this problem. So a way that we could approach this problem where we need no label variants to start with. And instead, we're going to use principles of evolution to infer variant labels. So there have been several methods previously that used tree structure to try to do variant calling in single-cell DNA. The idea is that a phylogeny constrains what mutations can be in which cells, and thus you could simultaneously try to fit a phylogeny and identify variants in cells. And these work generally fairly well on targeted whole exome data, but they do not work at all, essentially, for the low coverage data we're dealing with. They essentially expect up to maybe 30% sparsity instead of 95, 99%, which is what we have. But instead, I think we can actually do something simpler. We don't need to know what the phylogeny is. All we need to know is actually that there exists a phylogeny. And just that existence can allow us to make statements about which mutations agree with a potential phylogeny. And so just to back up a little and make sure we're all on the same page, I'm going to start with a little bit of vocabulary. I'm going to be talking about both single nucleotide variants and copy number aberrations here, because as I've said, we can detect copy number aberrations in this data with fairly high accuracy. The copy number profile of a cell is the vector of copy numbers at each position in the cell, so it's genome-wide. And I refer to a clone as a group of cells that has the same copy number profile. Then an SNV is said to be clonal if it's present in all cells in the clone, and it's subclonal if it's present in some cells of the clone, but not all. Given that vocabulary, we make two evolutionary assumptions. And these assumptions don't come from nowhere. They're actually completely consistent with a lot of recent methods that have been developed to do joint SNV and copy number phylogenetic models. And those assumptions are first, that single nucleotide variants occur only once during the evolution of a tumor, and they never back mutate. they may only be lost if there are deletions in the genome, essentially. And the second is that each copy number profile, so that the vector across the full genome evolved exactly once. Essentially what this translates to is that if we think of a clone in our phylogeny, it's a connected component. Given just those two assumptions, we have this nice property that pops out, which is copy number clones cannot share subclonal single nucleotide variants. That if we have two distinct clones of phylogeny, then they may have mutations that are shared and clonal by both of them, or private to each of them, or even this subclonal-clonal combination. But we'll never see mutations that are subclonal in both clones as long as these assumptions are met. When we go back to the tumor that I showed you earlier, however, we see a whole lot of variant calls that are subclonal in multiple clones. And the natural question is are all of these artifacts, or are they potentially violations of some of these assumptions? So what we did was we used a model to identify a confident set of shared subclonal mutations in this data and a confident set of shared clonal mutations, and we compared the properties of these two sets. What we find is that these shared subclonal mutations are fully consistent with what we would expect to see in artifactual variant calls. They have lower read lengths, lower mappability, higher numbers of mismatches, and so on. So we believe this is a reliable way to identify a set of artifacts in our data. So we tie these ideas together into a method that we call article for artifact color, where we use this key idea that shared subclonal SMBs are likely to be artifacts. And we use that to identify this confident subset of artifacts, as well as our confident reals, which are shared subclonals. We extract a number of read and genomic features, and then we could use this to train a classifier to remove artifactual SMBs. The nice thing about this classifier is actually because we can train it on any data we have, we can develop models that are specific to individual patients or cohorts or sequencing centers or whatever combination where we might expect to have slightly different error profiles. What we find is that when we compare against deep SVR, as well as several other methods that people use to do variant refinement, filtering by allele frequency, or taking consensus between multiple variant callers, that essentially most of the other callers or most of the other refinement methods have fairly high precision to start with, but the precision starts to drop off fairly quickly at like 60–70% recall here. Article, which is this purple line here, is able to retain much for 10 to 20% more recall at the same precision level. And we think this is because we are training it to pick up the specific error profile on this data here. Interestingly, what we find is that when we then map this back to copy number clones in our data, so this is just a mutation matrix. Each row here is a single copy number clone from our data, and the bottom here is presence and absence in the bulk. That in 5 of our copy number clones, we actually have considerably higher precision than we would see in that plot. And there are just 2 where the precision drops. So it might be that our actual precision is actually higher than what's represented here. It's just that these clones aren't necessarily shared by the 2 samples. So I'm going to just wrap up with an example of how this is changing the types of analyses that we're now able to do on this data. So I'm going to talk a little bit about mutation signature analysis. I think there was a talk about this yesterday, but just to recap, a mutation profile from a tumor is a summary of the types of substitutions that are present in the set of variant calls. They're frequently broken down by trinucleotide context, which is what we see here. And what I'm going to do is, for a cohort of patients, I'm going to compare the bulk profile that we observed for that patient with the DLP profile, the single cell profile, both before and after filtering. What we find when we do that is that, so have 62 samples here, and in 56 of the 62 samples, the similarity between the bulk and the filtered data actually increases, and oftentimes it increases to almost complete agreement. I'm going to focus a little on one single patient here, where we have a good considerable increase in our similarity, and show that we actually find something fairly interesting here. So we used SBM clone, which is a method for inferring cancer clones from ultra low coverage data based on SNVs. This was actually presented at HiTSeq 3 years ago. And we used it to cluster the SNVs in this data and identify clones. Before filtering, SBM clone essentially picked up no signal in this data, but after filtering, it's able to identify what we could see as a nice block structure, which is consistent with what we'd expect in a real clonal structure. And interestingly, what we find is that when we look at different clones, we see slightly different mutation profiles. So the ancestral is up top. These are things that happened early in the evolution of the tumor. Here we have a set of mutations that's private to clone 1, and then a set of mutations that's private to clone 3, or clone 4, I guess, actually. And what we could see is that, especially in this subset of C to G mutations, that there is an increase in signal here, and that is fully consistent with an APOBEC mutagenesis signal. So this is one of the most common mutagenic processes that we see across cancers, but it's actually relatively uncharacterized in ovarian tumors. And we see that there's, a very distinct increase of this in one of the clones. So this is now my single-cell RNA slide. We wanted to investigate why this signature might be enriched in clone 3, and we actually had conveniently matched single-cell RNA data from the same tumor, and thought, maybe we could find some phenotypic differences. So we used tree-align, which is a DNA-RNA integration method based on copy number, essentially, to match our DNA clones to our RNA data. And what we find that when we do that is that in clone 3, which has this APOBEC signature, there is lower expression across many repair pathways. Most interestingly, for the purposes of the APOBEC signatures, for base excision repair, which is the repair pathway that's primarily responsible for correcting these APOBEC-induced lesions in the data. So this points to a potential cause for this change in mutational processes. And it points also potentially to interesting properties that could be useful when we start talking about treatment, as tumors that are less able to repair damage may be more susceptible to certain types of chemotherapies, for example. So to wrap up, we have this method that we're very excited about for doing variant color refinement from single-cell DNA sequencing data, where the main benefit is we really require no manual labeling or the other input training data. We can infer a set of artifacts from the data alone. And this enables it to be highly customizable, and it's really opening up the types of analyses that we could start to do with this data. So to wrap up, I'd like to thank everyone that had a hand in this, especially my advisor, Saurabh Shah, and Matt Myers, who's done a lot on this, and patients and their families, especially, for donating this data.
That was a really nice talk. Do we have any questions? Oh, we have one up here, and then I'll grab you. Oh, yeah, for the people online.
Yeah, thanks a lot for the talk. Greetings for a fellow single-cell DNA-seq person. I feel your pain. And my question is, you probably also know that single-cell DNA-seq gets pretty much infinitely bad. And so what's the lowest coverage do you expect? Do you have any limit to how bad the coverage can be for your method to work?
So on this data, where we're typically getting usually about 0.05x coverage, I mean, to train the model, I think we need probably at least 10 variant reads to be able to call something confidently shared subclonal. So to train the model, we need reasonably good quality or a lot of cells. But then to apply the model, we've had success going down to as low as 80 cells in terms of filtering out artifacts.
Okay, thanks. Can I ask a quick second? Did you compare your results with the clonal, if you just do pseudo bulk per clone? And yeah, did you compare your results with the clonal events in this case?
We did. We find that actually it does a bit worse because the pseudo bulk, if we do pseudo bulk per clone coverage, the coverage is much lower. So variant callers just, really, they just call less, essentially, when we do that.
Makes sense. Okay, thanks a lot.
Thank you so much for the amazing talk. I have a question. What do you think, or what are your comments regarding like applying Articule to the for SMBs detected by bulk RNA-seq and single-cell RNA-seq?
So we're actually working on trying to get it working on single-cell RNA-seq. Kind of like all the principles here apply to almost any single-cell cancer data. The problem is that in data where we have like lower genome coverage, we need usually more samples just to get enough SNVs to train the model. But we're seeing some promising results, especially in single-cell ATAC data, actually, but single-cell RNA as well.
Okay, wonderful. Thank you so much. And another question is that I know that when you detect SMBs by RNA-seq, you don't catch them all. Like, for example, if you are doing whole exome. And so my question is, even if we have these tools, we will still have to integrate these results with exome sequencing.
Yep. Yeah, absolutely.
Oh, okay. Thank you so much.